All right, to start out, we are going to be talking about the small intestine and the growth of the small intestine. So, the small intestine is this organ that's pretty remarkable in terms of its renewal because the entire small intestine regrows every 5 to 7 days. So, and that's not entirely true. There's cells that last longer than that. But overall, the small intestine is made up of a lot of enterocytes. So, a lot of these cells that make up what are called the crypts or the villi of the small intestine are made up of these cells called enterocytes. So, each one of these guys, these little kind of light brown guys, are called enterocytes. They're the cells that are involved in the absorption of nutrients within the small intestine. And they have other functions. They house a bunch of enzymes on the surface, and they facilitate the movement of nutrients into the cell, and then the packaging of those nutrients from the space within the cell into interstitial space on the other side of the cell. So it's about absorption of nutrients. But a lot of these cells slough off, so they kind of fall off. And you can see here there's apoptotic cells where these cells die and they end up kind of breaking off. And so the small intestine is involved in a lot of renewal because cells are falling off, but I have to renew those cells through the action of stem cells, what are called intestinal epithelial stem cells. So these stem cells have to divide pretty rapidly and form a bunch of transit cells that also divide in order to replenish the barrier of the small intestine, replenish these cells that are continually falling off. And so the small intestine renews itself about every 5 to 7 days. And about in every day, upwards of 10 billion cells are lost. And that's about 30 grams of cells a day total are lost by this movement of cells, of these cells towards kind of the tip of these villi, sloughing off, becoming apoptotic, which means that they're kind of in a controlled cell death, and then breaking apart within the small intestine. And in a full year, that's equal to about 3.5 trillion cells that are falling off, but also being produced by these stem cells. So these stem cells have a pretty remarkable job in terms of the amount of cells that they're required to produce. And that 3.5 trillion cells produced per year is about 10% of the entire body's cell count. So our body is composed of eukaryotic mammalian cells. There's about 37 trillion cells, but that does not include the microbiome, which would add significantly more cells both on the skin and within the gut. But overall, we have 37 trillion cells in our body. And in a full year, there's about 3.5 trillion cells produced. So these are called the villi. They're these big projections in the small intestine that increase the surface area for the absorption of nutrients. But there's different cell types within the villi. A lot of these cells right here towards the tip of the villi are called enterocytes. They're the cells that actually line the gut and absorb nutrients. But there's other types of cells, like these purple guys labeled here, are called enteroendocrine cells. They're cells that are detecting nutrients or the presence of chemicals in the gut and releasing hormones or signal molecules into circulation. And then there's these cells right here, smooth muscle cells. There's macrophages, there's dendritic cells, so cells that are involved in the immune system. There's goblet cells that facilitate the production of mucin or mucus. So you can see all this stuff right here. All this stuff is labeled as mucus, which plays a role in the interaction with commensal bacteria, but also bacteria that are not commensal, potentially parasitic. So it plays a role in forming the boundary between the self and a space within the organism that's outside of an epithelium. And so if I go down here, one of the constraints that the small intestine has to deal with is its ability to produce adequate numbers of enterocytes. Because we're losing so many enterocytes, a lot of enterocytes are breaking off. I'm losing a ton. And these stem cells that reside at the bottom of what are called crypts are responsible for producing a bunch of stem cells. And these stem cells are able to divide continually. They're what are called multipotent stem cells. So they have the ability to produce other cells that are involved largely in just this sort of tissue type that's involved with the small intestine. But there's other types of stem cells. There's what are called totipotent stem cells that can become any sort of tissue. So they can become tissues involved in the formation of an embryo, which include ectoderm, mesoderm, and endodermally derived tissues. But they can also become tissues that form the placenta, so extra embryonic tissues. So these can kind of just become everything. Pluripotent stem cells are a form of stem cell that can become either the ectoderm, the mesoderm, or the endoderm of an embryo. And each of those tissues can give rise to a large amount of cells. But pluripotent stem cells cannot become cells that are associated with extra embryonic tissues like the placenta. So these guys can kind of become everything. These guys are a little bit more limited in terms of what they can become, but they can functionally differentiate into any tissue that's associated with an adult organism. So ectodermally derived tissues, mesodermally derived tissues, and endodermally derived tissues. But multipotent stem cells are a little bit more limited than the pluripotent ones. They can only really become tissues associated or become cells associated with a tissue or an organ. So these stem cells that are kind of sit at the very bottom of these crypts are what are called multipotent stem cells. So they can become cells like these enteroendocrine cells. They can become goblet cells. They can become transit cells that facilitate the further division upwards as you're moving up into these crypts. But they're limited where they can't become like a nervous tissue, for example. But this is a diagram that kind of illustrates how signaling, so there's a lot of signaling that occurs towards the base of these crypts. And then from the base, as they move upwards, there's a different signaling environment. So you can see here, towards the bottom, there's kind of more of a signaling environment that's associated with proliferation, so the division of stem cells, forming a stem cell and usually a transit cell. And then as you move further upwards into these villi, there's a signaling environment that's more associated with differentiation. And differentiation means that those cells are becoming specialized. So they're becoming enterocytes, they're becoming enteroendocrine cells, and then they can't really become anything else. They're stuck to that fate in terms of the type of cell that they're going to be. And so there's different signaling pathways associated with proliferation, like the Wnt pathway, the Notch pathway, the epidermal growth factor pathway. And then there, as you move upwards towards differentiation, there's more of a signaling pathway associated with keeping cells the same instead of proliferating, which is bone morphogenic proteins and transforming growth factor beta. Those sorts of signaling pathways play a role more up here, whereas proliferation stuff plays more of a role down here. And so there's a couple different cell types associated with the small intestine. And this figure may not be entirely accurate because I've seen some evidence that enterocytes make up upwards of 90% of the cells in the small intestine. But you can see here that there's certain transcription factors associated with the production of enterocytes that are different from the transcription factors associated with the production of goblet cells, same as Paneth cells, same as enteroendocrine cells. So there's a different signaling environment, which ushers on a different environment of transcription factors, which will then allow for the transcription of genes and the eventual translation of those genes into functional proteins that causes the differentiation of these cells and causes kind of a variety of different cells to be present. But overall, this diagram shows the function of these cells. So enterocytes are associated with forming the barrier of the small intestine, and then they're also associated with nutrient absorption. So on the brush border of the small intestine, which is like this little barrier right here that makes up the enterocyte, there's a lot of enzymes that facilitate the breakdown of nutrients so that I can absorb those nutrients into the cell and then move those nutrients from the space within the cell to the interstitial space on the backside of the enterocyte so that it can eventually move into circulation or potentially the lymphatic system if we're discussing chylomicrons. But each of these different cell types has a different function, but it has a different transcription factor environment, which allows it to differentiate and function the way it does. And so there's an issue with the small intestine, which is where if each of these stem cells is continually dividing and replicating, and we're growing an entire new small intestine about every 5 to 7 days, which is equal to about 10 billion cells per day that are lost but also have to be reformed. The issue is that every time a stem cell divides, it increases the likelihood that a mutation is introduced. And if a mutation is introduced in a stem cell, then that stem cell could potentially produce a population of cells throughout its life cycle that have that mutation and then can give rise to a faulty gut barrier or give rise to cells like these goblet cells or Paneth cells or enteroendocrine cells that kind of lose their function, which can introduce a lot of pathologies within the gut. So regulating mitotic age is extremely important. And cells do that by doing something like this, where a stem cell, let's imagine this is our stem cell right here, will divide and produce 2 daughter cells. One of those daughter cells will renew itself to become a stem cell, whereas the other daughter cell can then become what's called a transit cell, which can then go and divide, and each one of those cells can further divide, and that decreases the mitotic age versus something like this first example, where a stem cell divides. One of those divisions has to renew the stem cell, and then the stem cell differentiates straight into like a differentiated cell. And if it does that many times throughout its life cycle, you're introducing more and more cell divisions, which can increase the likelihood of a mutation occurring. So overall, there's some issues regarding the development of the small intestine over a long period of time. Because we're long-lived mammals, the small intestine has to regrow itself many times throughout the life cycle. Remember, 3.5 trillion cells are produced per year. So if you're living upwards of 80 years, you're producing a ton of cells and you're increasing the mitotic age of these cells a lot. So the body has many different means to decrease the mitotic age, and one of those means is through the usage of transit cells. So this is our stem cell right here. These blue guys are transit cells. Each one of those transit cells can undergo divisions. So each transit cell can divide further to produce differentiated cells, which will decrease the mitotic age here. Thanks, appreciate it.